Macromolecules are small molecules coming together to make a larger molecule. Macro means large, so macromolecule is going to be a larger molecule. Most macromolecules are going to be made from smaller molecules, repeating units to make a polymer or a larger molecule. The smaller units are called monomers. The larger molecule is called a polymer. A polymer is a bunch of monomers put together. Carbohydrates. The monomer for our carbohydrates is a monosaccharide. The polymer would be polysaccharide. Common monosaccharides for carbohydrates would be fructose, glucose, and galactose. These are all 6-carbon sugars. Carbohydrates are used for energy and structure. In animals, energy storage is glycogen. This is stored in your muscles. Plants, energy is stored as starch. And oftentimes plants have a reservoir to store their starch. Potatoes would be a great reservoir of the potato plant storage. They store large amounts of starch within that actual potato, and it grows underneath the ground, storing that starch or storing that energy for the plant that comes above the ground. Carbohydrates can also be used for structure. In plants, cellulose is the main form of structure. Paper is mostly cellulose, and it's been said that cellulose is one of the most common biological molecules on the planet. If you were to take all the biological molecules and kind of stack them up, the largest stack is going to be that of cellulose. And in fungus and in animals, mostly anything that has an exoskeleton like bugs and crustaceans have a structure carbohydrate called chitin. Chitin is found in the structure of these organisms. Carbohydrates have a ratio of their carbons, hydrogens, and oxygens in a 1:2 to 1. The most common ratio for a monosaccharide is going to be C6H12O6. So for every 6 carbohydrates, we have 12 hydrogen and 6 oxygen, so C6H12O6. Fructose, glucose, and galactose all have that same formula, but you can see that they are arranged differently, so the way that the carbon, hydrogen, and oxygen comes together is a little bit different. If we take a bunch of these monomers, the monosaccharides, and link them together, bond them together, we're going to come up with the polysaccharide. Starches, cellulose are all polysaccharides. If we take a look here, we can see those rings forming together. This is a picture of glycogen. This is how your energy is stored within your muscles. You can see that it's a bunch of these repeating units linked together. Then we have lipids. Lipids is not a true polymer because it is not repeating units of monomers. It has 2 subunits. The 2 subunits we have are glycerol and fatty acid. So our subunits are glycerol and fatty acid. Lipids are going to be our fats, oils, and waxes. They are used as a part of our membranes, our cell membranes are phospholipids. They also create waterproof membranes. Earwax would be a great example of a lipid. Beeswax is also a lipid. When we look at the chain of carbons and hydrogens of the fatty acid, it's going to determine how it acts in nature. If we look at the chain, we can see that this top one has no double bonds between these carbons. So they're all single bonds in between the carbons of the fatty acid here. It has as many hydrogens linked in as possible. This is called saturated. So the carbons are saturated with hydrogen. Saturated fats are mostly going to be animal fats. They are solid at room temperature and there's no double bonds. Things like butter, meat are all examples of saturated fats. On a general whole, if it's really delicious, it's probably a saturated fat. An unsaturated fat has a double bond. There is not enough it is not saturated. It does not have hydrogens as many places as possible because here, instead of double bonding, it could have the hydrogens in there, but it's not. So it creates a kink. And because it kinks, it changes the shape of the molecule, thus changing the structure. Because it has that double bond, it changes the shape. And when you change shape, you change function. Function and shape play a very large role together in biology. Unsaturated fats are going to be liquid at room temperature. One reason why they're liquid is because the legs are kinked, and because they're kinked, it's hard for them to compress together. And because they can't compress, they stay liquid. Mostly are plant oils, so olive oil, corn oil, these are all going to be unsaturated fats. And it's because they have one or more double bonds. Polyunsaturated means there's going to be more than one double bond in that fatty acid. Lipids, especially the ones in that large picture, are often used for energy storage, so your fat. When you're done using, or when you've eaten too many carbohydrates and you're not using it on the cellular level, it's going to then transform into a lipid and stored as fat. Another category of lipids are steroids. Steroids are a large ring structure shown here. Cholesterol is an example of steroids. Testosterone and estrogen are all examples of steroid hormones. The steroids that you would think about that you would take to grow muscle are a precursor to testosterone. So they are related to this example here. Nucleic acids. Nucleic acids are our information molecule. This is what gets passed on between parent and offspring. The monomer of a nucleic acid is a nucleotide. The polymer would be the nucleic acid. Here we have a couple more elements added in. We have nitrogen and phosphorus added in instead of just carbon, hydrogen, and oxygen. This is the basic structure of a nucleotide. There are 3 parts. We have our phosphate group here. We have a 5-carbon sugar, and this could be either deoxyribose for DNA or ribose for RNA. And then we have our nitrogen base. And I don't know if you remember from prior science, but this would be the A, T, C, and G of DNA. When we have repeating nucleotides, we're going to be creating nucleic acid. This is a short piece of DNA. We have our nucleotides here. So here's one nucleotide, and here's another. And as you go down the chain, you can see that it is repeating nucleotides. DNA is a double strand. RNA is a single strand. Protein. Protein is a polymer of amino acids. Amino acid would be the monomer in this case. The elements here are carbon, hydrogen, oxygen, nitrogen, and sulfur. Protein has a wide range of functions. Some of them are speeding up the rate of reactions, acting as an enzyme, fighting infection, transport within and between a cell, and movement. Your muscles, the reason why your muscles move is because of proteins acting together. We have a basic structure of an amino acid. Every amino acid has the same basic shape. We have the central carbon here. On one side, we have the carboxyl group. On the other side, we have an amino group, which is NH2. And then we have this R group. This is where it varies. There is no element R. Here, we would replace this R with different groups. And if we look at the picture below here, you can see all the different types of R groups that can go into each spot. So these are all different amino acids. The way that protein comes together is similar. We start off with primary. Primary is simply a chain of amino acids. The amino acids chain together in a big long string. Then they either pleat or helix. They can fold back and forth, or they can spiral. That would be the secondary level of structure. Tertiary is where those pleats fold on itself. Here we have some spiraling, we have some folding back and forth, and some more spiraling. So we took that spiral pleaty structure and kind of wrapped it in on itself. And then the last structure is when we take several tertiaries and shove them together. This would be a picture of a quaternary protein where we take several tertiaries and make a larger molecule. We're going to take a look at enzymes. Enzymes are a protein that speed up the rate of a reaction. Enzymes are specific to the molecule to the reaction that they are assisting. The parts of a enzyme mediated reaction, we have the enzyme itself. In this picture here, this would be the enzyme. This is the reactants of that reaction. The reactants are the molecules that go into the reaction, and for an enzyme reaction, that's called a substrate. A substrate is a reactant for an enzyme-mediated reaction. Substrate will sit into the active site. The active site is the place on the enzyme where the reaction occurs. This creates the enzyme-substrate complex. Enzyme-substrate complex is simply when the enzyme and the substrates are sitting together reacting. The reaction occurs, the new product leaves, and then you can reuse the enzyme over and over again. As long as that enzyme keeps the same shape, it will be able to be reused. The minute that you change that shape, it will not function. There are several things that can change the shape of an enzyme. It could be pH, it could be salinity, it could be temperature. If you think about enzymes in your stomach doing digestion, the enzymes in your stomach need to be at a very low pH of a pH of 1. If you change the pH, you're going to change the shape of those enzymes, and you're going to have unfunctioning enzymes, and you won't be able to digest the food. Oftentimes, scientists use the lock and key model, or one key for every lock, and that key only fits in that one lock. So the substrate is like the key, and the lock is like the enzyme, where that one key can sit in the lock and do the reaction and then leave. We're going to watch a quick little video here.
Nature's microscopic workmen. Hi, my name is Henry Goddard, and this is a presentation on how enzymes work. An enzyme is a living protein inside our bodies that helps us speed up reactions. It does this by lowering the activation energy of the reaction. This is Milo. Milo is an enzyme. This is Milo's activation site. This is where a substrate will bind on and cause a catalyzation. However, only a specific type of molecule will fit this site. Oh, here comes one now. This is Ernest. When he sits on Milo, he will split into two separate molecules. And as you can see now, we have Pete and Bertrand, who are pushed out of the enzyme. Milo, as you can plainly see, is an enzyme designed to break apart substrates. Remember, Milo didn't start this chemical reaction. It would happen regardless of whether he was here or not. He just speeds up the reaction. Now, let's say we have another enzyme, Larry the enzyme. Larry will attract two separate substrates. Here they are, Sylvester and Orlando. Like Milo, Larry seats the two substrates in his active site. However, unlike Milo, Larry fuses the two together. He now pushes one single molecule out. Steve. This animation was brought to you by